Welcome to Biology Minds. Today we're going to talk about the cardiovascular system, also called the circulatory system, and it consists of the heart, the blood vessels, and the blood. So starting off with this unit, we talk about the blood, then we're going to get into the heart, and then we talk about the blood vessels. So although they are one system, we like to separate them like we do with everything in anatomy and physiology, but ultimately they are one cohesive unit. So first, we'll talk about the blood. It's specialized fluid of connective tissue. So when we talk about the different types of tissue in the body, if you were to categorize blood, it is technically a connective tissue, even though it's not what you typically think of when we talk about connective tissue. It contains cells suspended in a fluid matrix. So it transports a number of things. Mainly, we talk about oxygen and carbon dioxide. So obviously, we need oxygen for cellular respiration, and in doing so, we need to get rid of carbon dioxide. So we are constantly taking in oxygen and releasing carbon dioxide. In order for our cells to get that oxygen and get rid of that carbon dioxide, we need our blood, specifically our red blood cells. So even though we think about oxygen and carbon dioxide coming in and out of the body, we talk about the respiratory system, but really in order for it to go to the specific cells, we need that blood to move it around the body. It also transports nutrients, hormones. We talked about that previously. Those are our chemical messengers, and they don't have their own vessels to get the hormones to. The endocrine system doesn't have their own vessels in order to get the hormones to the different glands or the different target cells, different tissues. So they're going to use the blood vessels and the blood in order to move those hormones throughout the body for them to get to those target cells. Also, we're going to find immune system components, white blood cells in there, and then also waste products that we're trying to get rid of from the tissue or from the cells. Some important functions. Obviously, we talk about transport, also regulation of pH, which we'll talk about more when we talk about the respiratory system and the urinary system with the kidneys. Restriction of fluid losses at injury sites, so we need those platelets in order to form a clot, so that if we do get injured, we're not going to have as much fluid loss. Also, defense against toxins and pathogens. We're going to talk about our white blood cells and stabilization of body temperature. So we have whole blood. That is when we're talking about blood as a whole, it consists of plasma and formed elements. So in the plasma, you're going to have water, dissolved plasma proteins, and other solutes. So we typically think about plasma as being basically water. But then within that plasma, we have formed elements, which are the cells that we talk about, our platelets, our white blood cells, our red blood cells. So if we were to break down kind of how much or the composition of the whole blood, we're going to look at the plasma makes up about 55% of that whole blood, and the formed elements or the cells make up about 45%. Within that plasma, it's mostly water, 92%. So of that 55%, we see that 92% of it is water. And in the formed elements, we see that we have some platelets, we have some white blood cells, but really 99.9% of it is the red blood cells. So we need those red blood cells to constantly be moving that oxygen, that carbon dioxide around, and it is important to have them functioning properly. So as much as we want our platelets there to do their job and the white blood cells there to do their job, we always need red blood cells there being efficient in order to get that oxygen moving through the body and get rid of that carbon dioxide waste. Typically, we abbreviate red blood cells as RBCs. The anatomical term or the scientific term is erythrocytes. White blood cells, WBCs, and they are called leukocytes. And then you have your platelets, which are involved in clotting. Hemopoiesis is the process of producing these cells or these formed elements by myeloid and lymphoid stem cells. So if you hear about stem cells are the cells before they become a specific type of cell. So you hear about the research where they're trying to learn about stem cells so that we can implant them into people that need new tissues. And these type of stem cells, these myeloid and lymphoid stem cells are the precursors to these formed elements. They're what cells are going to eventually create the red blood cells, the white blood cells, the platelets. Fractionation is the process of separating the whole blood for clinical analysis. So when we look at nurses and doctors, what they do, they're going to take whole blood and they're going to separate them in order to analyze, how much plasma is there, how much formed elements are there. We know how much there should be, and then we can figure out exactly what might be wrong with this individual based on if any of these numbers are off, if any of these percentages, the amount of water in the plasma or the amount of red blood cells that are in the formed elements and so on and so forth. So there are some general characteristics of blood that we can also check up on. The normal temperature of blood when it's in the body is about 100.4 degrees Fahrenheit. That's normal. It is very viscous, high viscosity, meaning it's very thick, right? When if you ever have blood, you see that it's not, it doesn't run quite like water would. It's going to be thicker than that, okay? And it might run slow, it might stick to things. And it is slightly basic or alkaline. We say it has a pH between 7.35, excuse me, and 7.45, so it's a little alkaline. We have 7% of our body weight is our blood volume, and in males we have 5 to 6 liters, and in females we have 4 to 5 liters. In our plasma, makes about 50-60% of the blood volume. More than 90% of the plasma in the water is water. Extracellular fluids, you're going to have interstitial fluid and plasma, materials, plasma, and interstitial fluid exchange across the capillary walls, where we see water ions and small solutes. There are plasma proteins in the plasma, albumins, globulins, and fibrinogens. We see that albumins transport substances such as fatty acids, thyroid hormones, steroid hormones. You have your globulins, which are going to be antibodies, also called immunoglobulins. Transport globulins, which are small molecules, hormone-binding proteins, metalloproteins, apolipoproteins, lipoproteins, and you have your steroid-binding proteins. And then you have fibrinogens. These are the molecules that form clots and produce long insoluble strands of this protein called fibrin. Serum is the liquid part of the blood sample in which dissolved fibrinogens converts to solid fibrin. All right, that is important in clotting. And then other plasma proteins, you have about 1% of makes up about 1% of the plasma. Changing quantities of specialized plasma proteins. You have these proteins or these peptide hormones, which are normally present in circulating blood. Insulin, prolactin, TSH, FSH, LH. All of these are part of those plasma proteins. Some of the origins, more than 90% are made in the liver. All right, so if you're ever curious about what the function of the liver has, is a very important role in creating these plasma proteins, antibodies made by the plasma cells, and then you have, once again, these proteins or these plasma hormones made by endocrine organs. Red blood cells, 99.9% of, not the whole blood, but of the formed elements portion of the whole blood. And the big important piece of red blood cells is hemoglobin. It's the red pigment that gives whole blood its color and binds and transports oxygen and carbon dioxide. So we need hemoglobin in order to transport that oxygen through the red blood cells. Red blood cells, about in males, about 4.5 to 6.3 million. In females, they have a little bit less. You see that with females, they have a scheduled hemorrhage every month. They are going to menstruate every month, and it's going to bring down their red blood cell count every month, where males are going to kind of have a more even keel to the amount of red blood cells that are in their body at any given time. Doctors or practitioners are going to look at the hematocrit, which is the packed cell volume, or PVC, the percentage of red blood cells in the whole blood. So they're going to put it in a centrifuge, which is something that spins the blood and is going to separate it into different parts, into the plasma, into the red blood cells, the white blood cells. They're going to be able to look at this, and by looking at that, that's going to be an indicator to them their overall health for that individual person. So when we look at red blood cells, they have this disc-shaped, but they're kind of a concave inside, so it's kind of like a donut that is concave, but it doesn't exactly have a hole like a donut does, and that's where we say that it's thin in the middle and thicker on the edges. So here is a cross section. Obviously, if we looked at a whole red blood cell, it would look like this, and this would be this concave version, middle of this red blood cell. Lifespan is about 120 days. We like to say give or take 30 days. So it's not an exact science where it's exactly 120 days, but it could be maybe 90 days, maybe 150 days, somewhere in that time. We're going to see that red blood cells are going to have to be broken down, and we're going to have to create new. So we're constantly replacing these red blood cells as each one hits that say 120-day mark. We're getting rid of them, we're breaking them up and getting rid of them, and we're making new ones. So we're constantly making new red blood cells. When we look at the hemoglobin structure, it has this complex quaternary structure. So we talked about early in the year about proteins, how they have that primary, that secondary, that tertiary, and that quaternary structure. When we look at hemoglobin, this is a protein that is very complex, where it is this long chain of proteins, and the structure is very important, and the shape of the structure is very important. It's very complex. So when you think about people with sickle cell anemia, when they are creating their hemoglobin, they have the wrong recipe or the wrong instructions, and by creating that wrong protein, is not going to be shaped the right way, and shape is really what matters when we talk about the function of these proteins. So there are 4 globular protein subunits within this hemoglobin, each with 1 molecule of heme, and each heme contains 1 iron. So this is why iron is very important in your diet. Here this shows you the structure. Breakdown of biliverdin. Biliverdin is a green color. It's converted to bilirubin, which is a yellow color. We see that bilirubin is excreted by the liver. If it is not excreted, if it starts to build up, then we see a yellowing of the skin that we call jaundice. So when babies are first born, their liver is not excreting that bile, that bilirubin, the way that it should be, and we see that they become jaundice, and then you have to actually lay them in the sun, and that helps to create the excretion of the bilirubin and eventually get that baby to be back to the color that you want it to be. So, first-time parents freak out that their baby looks yellow, but ultimately it's going to go away. It's just part of being a newborn baby. It's converted by intestinal bacteria to urobilins and stercobilins. Iron recycling, so iron is removed from the heme, leaving biliverdin to transport proteins, which is called transferrin, to storage proteins, which is ferritin and hemosiderin. Erythropoiesis is what we call red blood cell production. So sometimes we'll say red blood cell production, but erythropoiesis is the real name for that. It occurs only in myeloid tissue, which is red bone marrow in adults, and stem cells mature to become these red blood cells. So we talked about that. Those myeloid stem cells. Well, those myeloid stem cells are going to mature, and they're going to become red blood cells. Hemocytoblasts are going to be the stem cells in myeloid tissue that divide to produce those myeloid stem cells, which are going to create the red blood cells and some of the white blood cells. And then lymphoid stem cells are going to be really what are going to make our white blood cells or our lymphocytes. Some stages of red blood cell maturation. We see that you have on day 1 you have proerythroblast, and then erythroblast is 3 different stages, where you have your basophilic erythroblast, your polychromatophilic erythroblast, and then your normal blast, and then they become reticulocyte and mature, and then they become the red blood cells. So our mature red blood cells are anucleate because we eject this nucleus, and that is part of this erythropoiesis. Some stimulating hormones in creating red blood cells. So your body's going to keep an eye on how much red blood cells we have, and we want to make sure that we have the right amount. And in doing so, we need to use erythropoietin, which is also called erythropoiesis stimulating hormone. It is going to stimulate the production of red blood cells secreted when oxygen in peripheral tissues is low. So your body notices that you are not moving that oxygen the way that you should. We have hypoxia, which is a low amount of oxygen in the tissue, low amount of oxygen in the body, and that is going to create that erythropoietin release and is going to create more red blood cells. So often due to disease, and it could also be part of high altitude. So if you go out to Colorado and you're in high altitudes for an extended period of time, your body has to keep up with it because it's harder to take in that oxygen. Your body has to work harder, and that's why you see that marathon runners are going to train in high altitude, so that then when they come down, they are actually more efficient and they are better runners or athletes when they are not in those high altitude, and they're better than if they were to train in a normal place. So we talk. Sometimes you hear about blood doping where people are putting extra red blood cells in. Well, you don't have to necessarily do that. You know, that's illegal. You shouldn't be doing it, but you don't even have to do that because if you train in high altitudes, your body's going to make more red blood cells. You're going to have that erythropoiesis, and you're going to be more efficient at transporting that oxygen throughout your body. When we talk about blood typing, okay, anytime someone's in an accident, they are going to have to get blood transfused into them, and we need to know what blood type an individual is so that there are not any complications with this. So blood has these surface antigens, which are kind of like red flags. They're like markers saying, hey, this is part of the body. The body knows that this is part of it, and they're surface proteins that identify cells to the immune system. And normal cells are ignored, and foreign cells are going to be attacked. So we want our body to know, hey, this is not foreign. Do not attack it. There's different blood types, which is determined by genetics. So presence or absence of these surface antigens, you're going to have either A, B, you could have both, you could have neither, and then you're going to have an Rh factor, which is either going to be present or not, and that is what gives us our positive or our negative. So we have surface antigen A, that would be A. All right, surface antigen B, that would be B. And then if we have A and B, it's AB. And if we have neither A nor B, then we're going to have type O. Agglutinogens, those are antigens on the surface of the red blood cell. So surface antigens, agglutinogens, these are the same thing. Screened by an immune system, plasma antibodies attack and agglutinate foreign antigens. So we have plasma antibodies, where if you have type A, then that means you have type B antibodies. If you have type B, you have type A antibodies. If you have type O, you are going to have both A and B antibodies, and if you have type AB, you have neither A nor B antibodies. So here this shows you in type A, if you have type A surface antigen A, then you're going to have anti-B antibodies. Type B, you're going to have surface antigen B, anti-A antibodies. In AB, you're going to have both A and B antigens, but you're going to have no antibodies. And then in type O, you're going to have no surface antigens, but you're going to have both anti-A and anti-B antibodies. And then to determine if you are A or if you are positive or negative, so if you're type A positive, type A negative, type B positive, type B negative, there is a different one that we call Rh factor, also called D antigen. You're either going to be positive or negative. So separate from your A's, your B's, your O, you're going to either be positive or negative, and that's based on this Rh factor. Only sensitized Rh negative blood has anti-Rh antibodies. So where this becomes a problem is when you have a baby that inherited or through genetics has a different Rh factor than you, or more specifically when the mother has Rh negative and the baby has Rh positive fetus. So problems develop during the first pregnancy because the fetal cells, either the maternal bloodstream, then and thus the mother's immune system is not stimulated to produce the anti-Rh antibody. So nothing's really going to happen with the first baby because there's a separation between the blood. But then when she goes into pregnancy, you're going to see that there's exposure to fetal blood cells occurs during that delivery when the bleeding takes place at the placenta and the uterus. And then the mixing of the fetal and the maternal blood can stimulate the mother's immune system to produce anti-Rh antibodies, since she didn't have them before, leading to a sensitization. So then when she has the next baby, you're going to see about 20% of Rh negative mothers who carry Rh positive children become sensitized within 6 months of delivery. So it's not going to happen with that first baby. It's not going to happen with that first delivery, but you're going to see that it's going to the immune system of the mother is going to start building up this immunity to those Rh positive that Rh positive blood. And then it's because the anti-Rh antibodies are not produced in significant amounts until after delivery, and the woman's first infant is not affected. So there's no problem with the first one. But then in the next one, you're going to see that now there's an issue. The mother has an immune response to that fetus having Rh positive blood. And what they're going to have to do is that your doctor's going to have to monitor this, and they're going to have to actually give them a specific drug in order for them to not have this reaction. So they use this drug called Rogam, given to the mother in weeks 26 to 28 of pregnancy and during and after delivery. And these antibodies destroy any fetal red blood cells that cross that placenta before they can stimulate a maternal immune response, because that sensitization, you don't want it to occur, because otherwise then it can have an issue with the baby is going to start fighting the baby's blood. It's going to reject that baby. So we don't want any issues with that. So with the second pregnancy, you have to make sure that your doctor is monitoring this. The doctor should know, hey, this woman was Rh negative. She had a Rh positive baby last time. She could have another Rh positive baby, and it's going to be an issue. We have to give her Rogam in this 26 to 28 week period of the pregnancy, as well as during and after delivery. And what really happens is if the opposing antibodies are going to attack the surface antigens, we see that they're going to lyse or break up the red blood cells. We call hemolysis, and that's not what you want to happen. So we want to make sure that we are going to type a person's blood and make sure that they do not have an issue when we are going to transfuse any blood. When we are testing for blood, we are going to look for any clotting that takes place if we were to add those antibodies to the blood. So we see that if we put in anti-A serum, well, and the person has type A blood, we're going to see clotting, right? They don't have any B antigens, so we're not going to see anything for B. And then if they are positive, we're going to see clotting for the anti-D or for the Rh factor. Same thing goes for B. If you see that there should not be any issue with A, okay, you're going to see that there is with B and there is with positive. That's what makes it B positive. Here we see all three of them. That is going to give us our AB positive, and then if we see no response, that is going to give us O negative. So any of these are separate from the A and the B. So we can see that if this doesn't clot, then it's going to be A negative because we have our A, and this would make it negative. Same goes for these next two. If this one showed no clotting here, no clotting here, and then there was clotting here, it would be O positive. White blood cells, also called leukocytes, so we call them WBCs, sometimes we call them leukocytes. They do not have hemoglobin, and they have a nuclei and other organelles, and they function to defend against invaders or pathogens, removes toxins and waste, and they attack abnormal cells. They circulate around the body. Most white blood cells are in connective tissue proper, like we talked about in the 4th chapter when we talked about different types of tissue, different types of connective tissue, and then they're also in lymphatic system organs, but small numbers are actually in the whole blood, about 5,000 to 10,000 per microliter, very little compared to the amount that we find in the connective tissue proper and the lymphatic system organs. So 4 characteristics of these white blood cells: they can migrate out of the bloodstream, they have amoeboid movement, they attract to chemical stimuli, and some are phagocytic, meaning they are macrophages. And they are going to really engulf or consume the foreign bodies or the foreign cells or the bacteria. And these are neutrophils, eosinophils, and a big one is monocytes. Monocytes are very important. So types of white blood cells, we have neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Lymphocytes make up about 20 to 40% of the circulating white blood cells, are larger than red blood cells, migrate in and out of blood, and they're part of the body's specific defense system. So we see that T cells, they're going to be cell-mediated immunity. They're going to attack foreign cells directly. We'll talk more about T cells, B cells, natural killer cells as we move into the immune system or the lymphatic system. The differential count and the changes in the white blood cells are going to help to detect if there is any inflammation, any infections, and any allergic reactions. We actually keep an eye on CD4 counts for HIV patients, and this is going to help us to understand the relative numbers of white blood cells. How are the white blood cells doing in the body? And that's going to be an indication to the practitioners how the progression of HIV into AIDS is going. So if your CD4 counts are staying at the correct level, then we know that it is doing well. If we see that they're plummeting, then it's an issue. That means that HIV is going to become AIDS very, very quickly. So some disorders that we talk about when we talk about white blood cells, leukopenia is low white blood cell count. High white blood cell count is leukocytosis, and then an extremely high white blood cell count is leukemia, that we think of as cancer. White blood cell production. All blood cells originate from hemocytoblasts, which produce progenitor cells called myeloid stem cells, lymphoid stem cells. So we talked about those. These two cells are what make all white blood cells. They make our neutrophils, our eosinophils, our basophils, our monocytes, and our lymphocytes. So this shows you a diagram of how we have our hemocytoblasts, which are going to then either become myeloid stem cells or lymphoid stem cells. Our lymphoid stem cells are then eventually going to create our lymphocytes, where our myeloid stem cells are either going to become our proerythroblasts or our myoblasts or monoblasts, and then our proerythroblasts are going to eventually go through a number of phases and become our red blood cells. Our myoblasts are going to eventually become our basophils, our eosinophils, or our neutrophils, and then our monoblasts are going to become our monocytes. Platelets are cell fragments involved in human clotting system. They circulate for about 9 to 12 days and are removed by the spleen. So if you're ever curious what the spleen does, that's going to be breaking down our thrombocytes or our platelets as we create new ones, and 2/3 are reserved for emergencies. Platelet cells, we have platelet counts. We have over 150,000 up to 500,000 per microliter. Thrombocytopenia is a term for a low platelet count, and thrombocytosis is a term for abnormally high platelet count. So three functions. We talk about release important clotting chemicals, temporarily patch damaged vessel walls if you have a cut or a break in those vessels, and they reduce the size of a break in a vessel wall. Platelet production is called thrombocytopoiesis, occurs in bone marrow. Megakaryocytes are giant cells in the bone marrow, and they manufacture platelets from cytoplasm.